Okay, so we're going to go over chapter 3 today in class. So overview of chapter 3, you have definitions of cells, things about permeability, membrane transport, concentration, solutions, organelles, historical persons, what they're known for. Okay, that's probably going to be in the full notes, so we want to make sure that you look at the full notes. All right, so we have concepts of cell theory. It's also in the full notes. Differences between prokaryotes and eukaryotes. Definition of characteristics of functions of the cell membrane, membrane proteins, these different parts of the cell, different cell processes for membrane transport, active, passive transport, endocytosis, exocytosis, diffusion, osmosis, phases of mitosis in the cell cycle. You need to know how many cells you have at the end of mitosis. You need to know what factors affect diffusion rates. You need to be able to answer a potentially short answer question about transcription and translation. We'll talk about what those are today and next week. You could also have to look at a sequence of nitrogen bases on a nucleotide, on a template strand of DNA, and be able to write the corresponding nitrogen bases of mRNA. All right, again, you haven't learned about mRNA yet, but you will this chapter. Okay, bonus, if you're given nitrogen bases to the sense strand or the template strand of DNA, you'd be able to write the corresponding nitrogen bases of messenger RNA codon, nonsense strand or your coding strand of your DNA, and your transfer or tRNA anticodon. Okay, so that's kind of what we're going to be going over, all right, in this chapter. So I'm going to go back to the modules here. Scroll down to chapter 3 or week 3. All right. Let's go ahead and pull up the full notes this time. All right. Let's see. All right, so a cell, again, you're basically yes, ma'am. Thank you. Let's go ahead and get everybody else in here. I think it's a good time to do that. Thank you for the reminder. All right, we'll wait for them to get in so we can get started. All right, so let's start with chapter 3. So the cell, you have different levels of organization, right? So you have, your molecules, your organelles, but the point at which we consider life to occur is at the cellular level, okay? So some important people that you need to know about, Robert Hooke, and he described chambers in cork tissue, like, a little cork, piece of wood or plant, called them cells. That's the first time something was called a cell. Schleiden had the idea that all plants consist of cells in them, okay? And Schwann said that all animals consist of cells, okay? So 3 really important people that you'll want to know, okay? That could potentially show up as like a matching or something like that. But you'll definitely want to know that because it was on your review sheet. All right, contemporary cell theory is also on your review sheet. So cells are the building blocks of all plants and animals. Okay, important foundational concept. Cells are made when other pre-existing cells divide. Another big concept. Third concept, cells are the smallest unit that can perform all your vital physiological functions. Okay. Fourth, each cell is going to maintain homeostasis at the cellular level. What's homeostasis? You might remember? Balance, equilibrium, right? cells need to be in equilibrium. Fifth, homeostasis at the tissue level, the organ level, organ system level, and the organism level. It's going to reflect the combined as well as the coordinated actions of cells, okay, working together. The levels build on each other. All right. Let's talk about cells. So cells, the study of cell structure and function is cytology. Cyto means cell, ology is study of. Now, what kind of cells are there? They're prokaryotic and eukaryotic cells. This was also one of those things that was on that review sheet for chapter 3. You have prokaryotic cells and eukaryotic cells. The prokaryotic cells are bacteria. Your other cells, like your human body cells, your plant cells, your protocells, are eukaryotic cells. So one of the ways that they differ is in a nuclear membrane. Prokaryotes lack a nuclear membrane. Eukaryotes have one. All right. Prokaryotes lack most organelles. Eukaryotes have them. They have many organelles. Prokaryotes are small relative to eukaryotes. The eukaryotes are larger than the prokaryotes. In terms of their DNA, prokaryotes just have one circular strand of DNA. Eukaryotes are going to have many, many strands of DNA, and they're going to be in the form of chromatin. And your prokaryotes have small ribosomes, which have a different structure from what you see in eukaryotes. And in eukaryotes, you're going to see them roaming free, or you're going to see them attached to the endoplasmic reticulum. All right, a little bit about cellular anatomy. Two types of cells in the human body. You have sex cells or germ cells, and you have somatic cells. Okay, your sex cells are your reproductive cells or sexual reproductive cells. Your somatic cells are your non-sex cells. They're involved in forming the rest of your body. Your somatic cells, again, are your body cells. Basically, any cell other than a germ cell is a somatic cell. So basically, your only sex cells are your sperm cells and your egg cells. So you also, in the sex cells, would have anything from what we call primordial cell to mature gamete. So basically, anything that's developing into a sperm or an egg cell is a germ cell. All right, so your body's got 100 trillion cells in it. Okay, lots of cells. So everything we do is based on cells working together. Now, we said there's two basic cell types, somatic and sex cells, but to be more specific, inside of those categories, you have over 200 different types of cells. And most cells are going to have the same parts for the most part. And in your E-text, if you look at table 3-1, you can see that. Okay. Well, let's start with the cell membrane. Your cell membrane is going to be surrounded by what we call extracellular fluid. Extra means essentially outer, okay? So it's fluid outside the cell. All right. Your cell membrane, also known as a plasma membrane, forms your boundary of the cell. So what does the plasma membrane do? Well, it helps with isolation. So it forms a barrier, separates the inside of the cell from the outside of the cell. Regulates exchange with your environment. So it allows ions and nutrients to enter the cell, allows waste to get out of the cell, allows you to release secretions as well. Cell membrane is sensitive. It reacts to changes in the extracellular fluid. Remember in chapter 2 we learned about pH, okay, acids and bases. So it reacts to changes in pH, also reacts to changes in temperature. And it does that through receptors on the membrane. And those receptors, it's kind of like anybody have a satellite dish at home or cable box or thing on their roof or anything like that? Okay, it's a receptor. Okay, it picks up signals. So your receptors on your cell membrane do the same thing, and then they react to it, let you know what's going on outside, and you react on the inside of what's going on outside. Okay, it also provides structural support, makes your cells and tissues more stable. So these are good short answer questions, like tell me 3 or 4 functions of the cell membrane. Okay, it could also be multiple choice. Which of the following is not a function of the cell membrane? Okay, but when you see these underlined words, these are probably things you want to make sure you know, right? So always ask yourself, well, how might this look as a test question? Okay, and I'll try to tell you, but try to be thinking of that. And let's make sure we got our masks over our mouth and nose, everybody. Okay. I know they might not be the most comfortable to wear all the time, but we got to wear them. All right. So let's talk about the structure of the membrane. So it has a thickness between 6 and 10 nanometers. So what does it have in it? Obviously, it's going to have phospholipids. It's going to have lipids in it. Okay. The majority of the membrane is phospholipid. So it has lipids. Also has some protein in the membrane. Also has some carbohydrate in the membrane. It's known as a phospholipid bilayer. Bilayer just means it has 2 layers. Phospholipid means it's mostly phospholipid. The phospholipid molecules, remember in chapter 2 we learned about lipids. So you have hydrophilic heads, and what does hydro mean? Anybody remember? Water. What does philic mean? Love or like, right? So it likes water. Okay, and those are at the surface of the membrane. The tails that are hydrophobic, what does phobic mean? So they do not like water, so they're going to be in the inside of the membrane. So that means water, which is polar, and solute molecules can't cross through the lipid portion of the cell membrane. So polar molecules don't like nonpolar molecules. Which is an important factor in terms of isolating the cytoplasm from the fluid around it, extracellular fluid. All right, membrane proteins. So as the name suggests, they're proteins that are wedged in the membrane. They can be integral, which means they're part of the structure, or they can be peripheral, which means they're sort of attached on one of the sides of the membrane, but they're not really part of the membrane. Your membrane proteins can serve several roles. They can be anchors. And what do anchors do? Hold things down, right? Keep a boat in place. Okay, so they can attach to the cell membrane and stabilize the cell. Okay, you can also attach these cell membranes to the cytoskeleton. All right, it can serve as identifiers or recognition proteins. And they deal with how the immune system will recognize cells as normal or abnormal. It can also be enzymes. Okay, remember enzymes from chapter 2? They lower the activation energy of reaction and they can catalyze those reactions. Membrane proteins can also be receptors. They're sensitive to, and it should say ligands with a D, so Ligands. So if you have the notes with you, you might want to write that on there. It should be Ligands. All right, and they can bind to them and change the activity of the cell. Membrane proteins can also serve as carriers. Solutes can bind to the carrier protein, and the carrier protein can transport them across the membrane. Membrane proteins can also be channels, and they basically just form passageways where small molecules and water can cross through the channel, across the membrane. Okay, you see a couple different kinds of channels. They can be leaky channels, and they prevent water movement and ion movement all the time. Okay, so they can basically be open all the time, or they can be gated, which basically means certain conditions they're open, certain conditions they're closed. Okay? Does that make sense? Either they're open all the time or they're only open some of the time. And we'll talk more about gated channels in a later chapter. So the membrane, which is composed of your phospholipids and proteins, has certain characteristics. So it's fluid, it's not solid. Molecules move laterally. They can actually move across the membrane, or not cross it like from one side to the other. They can move along the membrane. Okay, so that, if this is the membrane, or this is the membrane proteins can move along that membrane. Okay, so they can slide over. The proteins aren't distributed evenly, so it's mosaic. All right, so anybody see mosaic tile, where you take lots of different tiles and mix them together? Okay, it's the same concept. So it's a fluid mosaic model. That's the way we describe the cell membrane. And your cell membrane composition can change based on reactions that happen or metabolic turnover. So the inside surface and the outside surface of the membrane don't have to have the same protein composition or lipid composition. They can be very different. Okay, we talked about proteins in the membrane. Let's talk about carbohydrates in the membrane. So I don't know if anybody remembers, we talked a little bit about a structure called a glycocalyx. Okay, so there's some sugar on the membrane. It can serve a role of protection. It can also help with lubricating the membrane. It can serve as an anchor. In some cases, it can help with motion. They can serve as receptors. Part of that glycocalyx can be sensed by the immune system. It can help you tell if a cell is a normal cell or it's an abnormal cell. And all of these characteristics are based on the genetics, okay, of the person, based on the person's DNA. Right. The levels build on each other. Okay, let's talk about permeability of the membrane. Permeability is just a fancy word that basically means how easily certain substances can get across the membrane. If a membrane is impermeable, nothing gets across it. If the membrane is freely permeable, any substance can cross over. But our membranes are selectively permeable. Okay, they can permit the passage of some materials and restrict the passage of other materials. Okay, selective. All right, so what I want you to do now, take a minute, look over the person next to you, and explain to that person. I give you a minute, explain those 3 different kinds of membranes. So then we'll let the other person go. Go. All right, let's talk about active and passive transport. So passive transport, no energy is needed to move ions across the membrane or molecules across the membrane. But if it's active transport, it's going to require energy, usually in the form of ATPs. So there's 4 categories of transport. It can be diffusion, filtration, carrier-mediated transport, and vesicular transport. So you need to know those 4. You also need to know the difference between active and passive transport. So let's start with diffusion. All right. So this just means movement of material from the area where the concentration is high to the area where the concentration is low. All right, so you're basically moving down the concentration gradient, which is just your difference between the high and low concentrations. Okay. So diffusion is going to happen until you eliminate the concentration gradient. So your goal is not to have diffusion happening forever. It's to get to a point where there's no concentration gradient. Then there's movement, but the concentrations are going to stay the same at that point. All right, so what are factors that influence diffusion rates? That was also on your review sheet, okay? So distance, size of the gradient, molecule size, temperature, and electrical forces. So if you have those printed out or you have them on an iPad where you can draw on it, you might want to underline those. So with distance, the shorter distance you got to travel, or the molecules have to travel, the faster your diffusion is going to happen. All right, so like if I have to walk across from one side of the room to the other, it's going to take me less time to walk halfway than the whole way, right? Same concept. Size of the gradient, if you have a larger concentration gradient, you're going to have faster diffusion, okay? Because the difference is so high, it essentially creates more sense of urgency to move. Okay, size of the molecules. Ions as well as small organic molecules are going to diffuse more quickly than larger proteins. So If I wanted to move across the room, and I can carry, one book, I'll move at one speed, but if I have to carry a whole big stack of books, I'm probably not going to move as fast, right? Same concept. The temperature is higher, you're going to have faster diffusion. Why? Because temperature creates energy, right? Heat is energy. More temperature, more heat, more energy, molecules move faster. And electrical forces. So you can have concentration gradients, but you can also have electrical gradients. So negative charges on inside of the cell membrane can pull the positive ions from the extracellular fluid toward the inside. And so the net result of these chemical and electrical forces acting on an ion is called an electrochemical gradient. So you're looking at both the concentration gradient and the electrical gradient. So how does diffusion happen across a membrane? Two ways. You go through a channel in the membrane, some channel protein, or it can go through the lipid portion of the membrane. Those are basically the only ways you can go through. So osmosis, really what you need to know about osmosis is this diffusion of water. Okay, if you get that, you've kind of got the main point there. It is diffusion, but it's diffusion of water. If it's diffusion of anything else, it's not osmosis. So what are characteristics of osmosis? Well, you have diffusion of water molecules across the membrane. Okay, that's pretty obvious. And also it occurs across a selectively permeable membrane, like our cell membrane. It's freely permeable to water, but not freely permeable to solutes. So water can cross the membrane in some way, but most solutes or many solutes cannot. Water is going to flow across the membrane toward the solution. It's going to have a higher concentration of solute. Well, wait a minute, Dr. Edwards. I thought you said diffusion is movement from air high concentration to an air low concentration. We're talking about the water, okay? So if the concentration of solute is high, concentration of water is low. So the water is going to flow toward the high concentration of solute, okay? So osmotic pressure. So there's pressure related to this difference in water concentration. So it creates a force of movement of water. That's osmotic pressure. You also have hydrostatic pressure, and that's pressure of the fluid. And it can actually oppose the osmotic pressure. It pushes against the fluid, so it kind of resists osmosis. Another term you need to know is bulk flow. So when you buy things in bulk, what does that mean? You buy a whole lot of it, right? Like you go to Sam's or Costco or something and buy things in bulk. Bulk flow just has to do with large groups of water molecules moving across a membrane at once. Okay, so it's not just a molecule here or there, it's a lot of molecules moving. That's bulk flow. All right, you also need to know about osmotic concentration, or another term for that is osmolarity. So here you have the total solute concentration in what we call an aqueous solution. Okay, it's osmotic. It's one type of that solution, and that's iso means what? You might remember? Same. So in an isosmotic solution, your solution is going to have solutes that are in the same concentration as the cytosol in that cell. Okay, so basically extracellular fluid has the same concentration as the cytosol. In the hyperosmotic solution, the extracellular fluid is going to have a higher solute concentration than the solute of the cytosol of the cell. In a hypoosmotic or hyposmotic solution, remember hypo is low, the extracellular fluid solution is going to have a lower solute concentration than the cytosol. All right, you can also describe the cell membrane and osmosis and diffusion in terms of tonicity. Okay, so this is basically how these osmotic solutions affect living cells. So you probably heard these terms in lab: the isotonic, hypotonic, hypertonic. Isotonic, so there's no osmotic flow, and that's, I want to make sure it's clear. It doesn't mean that there's no movement. It just means there's no net movement. Okay, so there's still water moving, but the water moving in and water moving out are moving at equal rates. Okay, because the concentrations inside and outside the cell are the same. In a hypotonic solution, water is going to flow into the cell because the concentration of solute outside the cell is low. It's hypo, which means the concentration of water is high outside the cell, which means water is going to flow in. What's the effect of this? Can lead to something we call, in the case of red blood cells, hemolysis. Okay, hemo has to do with like hemoglobin. That's something you find in your red blood cells. Lysis means to break. So you burst, if you burst the red blood cell, it's called hemolysis. In a hypertonic solution, that means the concentration of the solute is higher outside the cell. Okay, and if the concentration of solute is higher outside the cell, it means the concentration of water is low outside the cell, which means water is going to flow out of that cell to the extracellular fluid. It means that red blood cell or other cell is going to shrink. Okay, and that's a state that we call crenation. All right, so what are fluid shifts? Well, again, because we're talking about water, it's osmotic movement in or out of the cells. Okay, again, we want to maintain homeostasis, so even with minor changes in the intracellular and extracellular fluid, solute concentrations, those fluctuations get eliminated quickly. Okay. But if you have system-wide changes in your body in solute concentration, that's obviously going to take more time to fix. Okay. What is filtration? Okay, if you have hydrostatic pressure, it's forcing water across a membrane. Okay, you're forcing it through a filter. If you have large membrane pores, your solute molecules can get carried along with the water. In the human body, you have filtration in certain places, like in the capillaries, right? So that your capillaries are designed to be able to filter materials into or out of the capillaries. You also have filtration happening in the nephrons, in the kidney, right? That's where you want to be able to eliminate waste, right? Okay, let's talk a little more about transport. One form of transport, so we talked about diffusion. Let's talk about carrier-mediated transport. And that just means you have integral proteins. Remember, those are proteins embedded in the membrane, and they're going to bind specific ions or specific organic substrates to get them across the membrane. Okay, they can't, these are molecules that can't go just straight through the membrane. They have to use a carrier. And these carriers have characteristics that are similar to enzymes, another type of protein. They're specific. Okay, carrier proteins just don't take anybody. Okay, they're designed for a specific ion or a specific organic substrate. All right, so one way to think about it is it's like carrier-mediated proteins are kind of like custom-fit clothes. Okay, they're designed for that person, right? So as opposed to just saying, anybody who wants to wear that can wear that. So saturation, so your rate of transport is going to be limited or controlled by how many solute molecules and carrier proteins there are available. So if you have a lot of carrier proteins, you have a higher transport rate because you have lots of options for these substrate molecules. If you don't have a lot of substrate, you're going to have a slow transport rate. But let's just say you have more substrate than you have carrier proteins available. And it's going to slow down your rate of transport, right? So if only 20 people can get on the bus, if you got 50 people waiting for the bus, it's going to take, 2.5 or 3 bus rides to get everybody where they need to go. So regulation is another characteristic of carrier-mediated transport. Carrier protein activity can be altered by binding of other molecules. So hormones can regulate transport across the membrane. So what does carrier-mediated transport move? Well, it's going to move things like single substrate molecules, okay, get them across the membrane. So you also have you can move 2 different substrates in the same direction. It's called co-transport. So some carrier proteins have spots for 2 different materials. Kind of like a vehicle has 2 seats. You can also have carrier proteins that move substances in opposite directions. We call that counter-transport or antiport. Okay, so the 2 seats don't necessarily have to be moving in the same direction. So you have different types of carrier-mediated transport. One is facilitated diffusion, the other is active transport. Okay, so facilitated diffusion is diffusion. So it's moving from an area of high concentration to low concentration. It just requires a facilitator, or some sort of carrier protein. So the materials or substrates will bind to the receptor site on the carrier protein. That carrier protein is going to adjust to that and change shape. That's going to allow those compounds to get transported across the membrane. Facilitated diffusion is great for compounds that are lipid insoluble, right? Because lipids can't go through that middle part of the bilayer. Things like glucose and different amino acids, they can't get across. It's diffusion, so there's no ATP, no energy used. Again, you're moving from high concentration to low concentration. Okay, so again, once you basically filled up all the sites on the carrier proteins, your rate of transport is basically doesn't increase at that point. There's no seats available. You hit your limit in your transport rate. All right, so let's take a minute, talk to your partner about facilitated diffusion and diffusion and osmosis. All right, let's see. Let's talk about active transport. So this is going to need ATP. You're moving ions or molecules across the membrane, but it's basically against or independent of the concentration gradient. Okay. So most of the time you're going to be opposing the concentration gradient in active transport. So one of the ways that you can do that is through ion pumps. Example of that would be carrier proteins transport ions like sodium, magnesium, calcium, potassium across the membrane. So many of these ion pumps will move a cation in one direction, okay? They only move one direction. And many of them actually move more than one cation at a time. So, you know, there's some either co-transport or anti-transport. And so, oftentimes it is anti-port. So you have an exchange pump where you move a cation in one direction and then another cation in the other direction. Example of that, sodium-potassium pump. Exchange sodium inside the cell for potassium outside the cell. And in your notes in lab, you should have a picture of the sodium-potassium pump. All right, secondary active transport. Here, the carrier transport is not using the energy, but the cell's going to need energy in a subsequent or second step to keep homeostasis. Sometimes you're removing another substrate at the same time. And your concentration gradient for one of the substances is going to give you the driving force you need for that carrier protein to transport the other substance. All right, so it can be involved with both co-transport or counter-transport. Okay, so what's the difference between active transport and passive transport? Big difference. What's the big difference? Okay, active transport requires energy. Okay, requires ATP usually. All right, vesicular transport, form of bulk flow. You're moving materials in and out of the cell through vesicles. They're basically spheres made up of essentially the same material your cell membrane is made up of. And we'll learn more about that when we talk about parts of the cell. Okay, it can be through what we call endocytosis or exocytosis. In endocytosis, endo means inside, right? So this is a process where the material gets transported into the cell using a vesicle. Okay, it's typically an active process. It's going to need ATP or energy. All right, so basically the contents of these vesicles, they take material from the side, basically outside the cell, and bring it into the cell, stay inside the vesicle. So it's three different types. You have receptor-mediated endocytosis. Just means that there's a receptor involved in sensing things on the outside of the cells. Okay, so you have specific receptors that look for specific molecules. Okay, and when they find those molecules, it causes the membrane to do what we call invaginate, which basically means if this is part of my cell membrane, if I have a receptor and I sense something, it's like, oh, we got to get that taken care of. So that membrane is going to start pinching in on itself and close up and go in the cell. Okay, so pinches off to form a vesicle. You also have Pinocytosis. Right, and Pino means drink. All right, so you have formation of these invaginations on your cell surface, and they close to fill or form vesicles that are filled with fluid. Okay, the drinking, cell drinking, cytosol. Okay, you also have phagocytosis. Phago means eating, so you have cell eating. All right, and this time you get invaginations that will enclose what we call particulate matter. Okay, this happens in certain specialized cells like white blood cells. And you might know what white blood cells are good for. Help you fight infection. Okay, so if they sense something is bad, they react to it. If this is a white blood cell surface, if they see something that you shouldn't be in the cell, it closes in on it, degrades it. Okay. And it does that through what we call pseudopodia. They're like, kind of like feet. Pseudo means sort of. Pods are kind of like projections. And so they're basically extensions of the cytoplasm, and they'll surround the object. And the cell membranes will fuse, make a vesicle on the inside of that white blood cell. Now, that vesicle will then meet up with the lysosome. We'll learn more about what that is. We talked about, if you had lab this week or last week, you probably learned about lysosomes. But the lysosome will degrade what's inside that vesicle. Okay, so that's endocytosis, bringing things in. Exocytosis is the reverse. Sending in exo means out, right? Outer. That's taking things out. So it could be good things in the case of secretory products, so things that you need to send to other cells. They could be bad, or they could be waste products and sending them out. All right. Okay, talk with your partner. Difference between endocytosis and exocytosis. Go. So Alicia, you can tell me. What's the difference? All right, other partner. Okay. Yes. Yes. So remember, endo is inside, exo is outside. So just help you remember. All right. Okay, let's bring it back. All right. So Exocytosis, just talked about that, so we're good there. Here's a concept I want you to know about transmembrane potential. Trans means across, okay, or different sides. So one side of the cells, outside is going to have a positive charge. Inside of the cells is going to have a negative charge. So you create a transmembrane potential. We also talk about what we call a potential difference, when the positive and negative charges are held apart on that transmembrane potential. Okay, it's another way of describing it. Okay, now the transmembrane potential, another way of talking about that, it's the difference, potential difference between two sides of the cell membrane. So potential difference is something you'll see a lot in physics when they talk about electricity. Transmembrane potential is kind of like a bio or cell biology application of that. All right, so when we get to muscle contraction and nerves and all that, we'll talk a lot more about this. But the potential difference between the two sides of a cell membrane in a living cell is considered negative 70 millivolts, okay? So that number may not mean much to you now, but like I said, when we get to muscle contraction, it will mean a lot more to you. All right, cell membrane. It's going to resist these electrochemical forces to try to move these ions in and out of the cell. All right, so the ions that are held apart, by being on the opposite sides of the cell membrane, are going to have potential energy. And remember, potential means not being used, right? Cell's going to use this potential energy for different reasons, like sensing information along the surface of a neuron. Okay, so we've learned about transmembrane potential. Let's talk about resting potential. This is a transmembrane potential in an undisturbed cell. It could be anywhere between negative 10 millivolts and negative 100 millivolts. And it actually takes energy to maintain this resting potential. Okay. So there's forces that would want to make the potential outside of that range. It takes energy to keep it in that range. All right, I think we talked enough about potential differences and all that good stuff. Let's talk about organelles. All right. So cytoplasm, start there. When we talked about the cell membrane, let's talk about the organelles on the inside. Let's start with cytoplasm. It's inside the cell membrane, but outside the nucleus. Two main divisions of the cytoplasm. You have the cytosol, which is the fluid, or intracellular fluid. It surrounds the other main portion, the organelles. So that cytosol is going to have the organelles in it, the nutrients, ions, other proteins, other waste products that are dissolved in it. The organelles are your cell structures, and they all have different functions. Okay, there's some specialization to the cell membrane, or there's some specialization to the organelles in the sense that different organelles have different functions. Okay, give the cell its structure, help you maintain your cell, and it's involved in metabolism, which is another fancy word for chemical reactions. So how is the cytosol different from the extracellular fluid? Well, the cytosol has a high concentration of potassium ions and a low concentration of sodium ions. It's the reverse. In the extracellular fluid, it has a high concentration of sodium ions and a high concentration of potassium ions. And that's really important in that sodium-potassium pump we talked about earlier. Another difference has to do with suspended proteins. Cytosol has a high concentration of suspended proteins. The extracellular fluid has a low concentration of suspended proteins. Your cytosol is going to have small quantities of things like carbohydrates, but large reserves of amino acids and lipids. Your extracellular fluid will have small quantities of amino acids and lipids. Okay? Your cells are the ones that are going to need those amino acids to make proteins, and they need those lipids to make energy. Your cytosol can contain things we call inclusions, whereas your extracellular fluid will not. And the inclusions are just masses of materials that are insoluble in the cytosol. Okay, our organelles can be described as non-membranous or membranous. Okay, if they're non-membranous, they're always in contact with the cytosol. So things like the cytoskeleton, ribosomes, cilia, flagella, centrioles, and the microvilli are non-membranous. Our membranous organelles have lipid membranes that separate them from the cytosol. So some of the ones that you may be more familiar with, like your endoplasmic reticulum, your mitochondria, your lysosomes, your Golgi apparatus, your peroxisomes, and if you're not, that's okay. Those have membranes. So let's start with the non-membranous organelles. So your cytoskeleton is composed of your microtubules, intermediate filaments, thick filaments, and microfilaments. As the name suggests, cytos is cells, skeleton is support, or structure, so it gives the cytoplasm strength, but some flexibility too. The microfilaments are thin strands of protein. They're usually made up of actin, very small diameter. They serve as anchors for integral proteins, connect them to the cytoskeleton. Give the cell additional mechanical strength. They also help to determine how thick the cytoplasm is, whether it's more fluid or more viscous. They're also involved in active movement. So particularly in cells that have to move or cells that change shape. And they interact with another protein called myosin. Okay, and we'll see that a lot when we talk about muscle contraction. All right, your intermediate filaments. Okay, they are larger in size than your microfilament. But they're going to be smaller in size than the thick filaments. Okay, that's why they're called intermediate. A little bit larger diameter. Okay, it gives, again, strength and stability. Okay, basically hold the organelles in place. And also basically keep the cell in position relative to other cells. All right, so thick filaments, again, these are the larger parts of the cytoskeleton, massive bundles of myosin. You only see these in the muscle cells. And why do you need these? You need these for muscle contractions. Your microtubules are hollow, made up of what we call tubulin. and they have the biggest diameter. How many and how far, where they're distributed in the cell will change over time as your cells get older. All right, and it's a primary component of your cytoskeleton. It gives your cell strength, rigidity, holds it in place. All right, so when these microtubules disassemble, allows for the cell to change shape. We have other intracellular materials that can move along that microtubule. They're also involved in cell division. They help form the spindle apparatus. It helps those chromosomes move to different sides of the cells so they can divide. Also helps in the structure of organelles like cilia, flagella, and centrioles. All right, so we're going to stop here. What I'd like for you to do is to work on your exit tickets, and let's see, we will pick up next time.